Laura Jardim from Brazil, covering the topic, What's New in the Genetics of Huntington Disease. After Professor Julie Stout from Australia will discuss the clinical continuum from pre-manifest to manifest Huntington disease. And at the end, Professor Anne Rosser from the UK will discuss the present and future of a multidisciplinary approach to Huntington disease management. At the end of these presentations, we will invite all of you to attend the live discussion that will follow. This session will also feature top abstract presentation. The top abstracts program highlights the best original research presented during the MDS Congress. Before we start, I would like to remind all participants that the materials presented in the MDS Virtual Congress are for educational purposes only and are not to be recorded or reproduced. Photos of your screen or any presented content is not allowed. All relevant faculty disclosures for accredited scientific sessions can be found online at the MDS Congress website. Please post your questions for the faculty in the chat feature. And finally, please do not forget to provide your feedback in the session evaluation. You will be able to find the link in the chat feature or in the scientific program tab on the Virtual Congress platform. So thank you for attending this session and the MDS Virtual Congress. Hope you enjoyed this session on Huntington disease.
So hello, everybody. I am very grateful to participate in MDS Congress. My task is to review with you what's new in the genetics of Huntington's disease. And well, we do have breaking news. As they are complex, I'm going directly to the point. So the breaking news was brought about by the discovery that the pure CEG tract inside the CEG repeat, expanded repeat, is directly related to the age onset in Huntington's disease. This is not as trivial as it sounds. Actually, this discovery takes on the dimension of a paradigm shift when it is combined with two other lines of genetic evidence. And the first one is the relationship between DNA repair system and age onset in Huntington's disease, while the second one is the evidence that there is neuronal mosaicism of the expanded CEG size in subjects affected with Huntington's disease. So when these three elements are brought together, they support a new explanatory theory for the late onset of neurological disorders due to repeat sequences and expansions, of course, the two stages hypothesis. In my opinion, the most emblematic image for this epistemological, I would say, great leap forward is the figure from the GEM-AD-HD Consortium 2019, but I will get there. Okay, so briefly and starting from the beginning, Huntington's disease is due to dominant expansions of a CEG repeat tract expanded on HTT gene. Huntington's is characterized by the presence of a movement disorder featuring chorea plus psychiatric changes and cognitive decline. Evidences allow us to state with certainty that in Huntington's disease, there is complete dominant gain of function related to a CG repeat expansion that is unstable. Until recently, the prevailing explanation for that was the slippage of DNA during replication, or in other words, assuming the need of cell divisions before that. But more recently, and also due to the new developments, the fact that the instability is also due to defects in post-transcriptional DNA repair, especially in post-mitotic cells, has come to be more appreciated. So what is the importance of this on pathogenesis of the disease? Of course, polyQ causes intracellular proteic toxicity. This is well established. This is a fact. However, later, evidence appeared on transcellular toxicity of polyQs, but I will not put my focus on that. I will have no time. Actually, the most important thing on the top of all that is the evidence pointing that the polyQ toxicity depends on additional continued intraneuronal expansion, questioning the CEG repeat size threshold to neuronal damage. So the neuronal damage, the threshold for that might be greater than we thought from the HD genotypes from the blood periphery. So Of course, in brief, we know that normal alleles produce proteins with up to 35 glutamines due to the CEG repeat, and they are okay. While pathogenic alleles produce huntingtins with 36 or more glutamines according to the genotypes in blood periphery. Okay. The expanded polyQ is prone to aggregation, localized in the nucleus, and is related to neuronal damage. And several distal events or effects due to polyQs were already described. And most of these effects, there are a lot of them, were raised from cell and animal models where the size of the polyQ needed to be larger than the estimated by the polyQ in humans, or 100, I would say, 100 or more in models versus the 40 or something that we found in the blood cells. This difference was mostly attributed earlier to interspecies differences. Other explanations might have been overlooked. However, since 2019, several researchers started to sustain that a step farther than polyQ damage is needed to disease to start. Further CEG expansions in neurons from the inherited CEG sites. And this has been called the two stages hypothesis. It's actually, I would say, a revival on a hypothesis presented in 2007 by Kaplan and co-workers. They said that to have a disease due to an unstable expansion, a person must inherit a large allele. This allele is prone to further expansions in somatic cells during life. As time passes, CEG expansion might cross a threshold associated with neuronal damage. And as a critical number of neurons are reached, then symptoms would start. However, in 2007, this hypothesis was supported only by a few papers reporting neuronal somatic mosaicism in Huntington's disease. And technical questions were raised at the time regarding this finding. Today, somatic mosaicism has taken the lead in theories of pathogenesis, however. How did it happen? Well, by the interrelationship between these three lines of evidence. And I will try to tell you the story of that. So everything seemed to take shape actually in 2019 after three major groups reported on the same discovery that the pure proximal CEG sequence in the expanded repeat was more strongly associated, I might say, with age onset than the estimated size of this expanded polyglutamine, as estimated by the CEG genotype in blood. The three papers reported that the rare variant of the CEG repeat with loss of interruption, this kind of variant, was associated with a strong anticipation in onset. So this variant is quite rare. It has been known since the 90s, whereas this is the canonical allele in us and in affected subject as well. We have this interrupting codon CAA. Of course, there is another rare variant with double duplication. It's important to stress that both codons, the CAG and the CAA, codes for glutamines. So from the point of view of the protein, it does make no difference. Well, one of the first papers, the GEM HD paper consortium found a signal near the HTT gene. And this signal was related exactly with these two variants, these two rare variants. What they saw, they found only 19 out of 9,000 HD subjects that carried the loss of interruption allele. And these guys have early onset from the mean ages at onset of the others, while the carriers of the double interruption have late onset on the disease. But most intriguing, the effect of the purity per se, the non-interrupted sequence, was larger than the estimated polyglutamine length. Can you see that? I think that this cartoon puts HD subject in a better way to explain. So all subjects here from the GEM HD consortium are presented according to their non-interrupted CEG repeat sizes. So for instance, and these guys here all have 43 CEG repeats in the pure sequence, either carrying a duplicated repeat or not carrying duplications at all. Yeah, so it means that these guys have 43 repeats, 45 repeats, and 47 repeats, codons, coding glutamine. If the timing of onset was determined by the polyglutamine length estimated by the CEG sizes, these guys with double duplication for identical